Welcome, you curious learners, to a new deep dive. Today we're embarking on a truly foundational journey, one that really underpins all of human health and development, conception and prenatal development. We've got a fantastic stack of material here, really laying out the incredible step-by-step process, from a single cell all the way through to birth. Our mission for this deep dive is to extract the most important nuggets, the aha moments, so you walk away with a clear, structured understanding of how it all begins.
Yeah, and what's truly fascinating here is how these early microscopic events lay the entire groundwork for human health and well-being. For anyone stepping into the healthcare field, understand this normal development isn't just, academic knowledge. It's the critical context for everything that follows. It lets you recognize when something might deviate from the norm, understand the potential implications, and ultimately provide better care down the line. So we'll trace this really intricate path from the formation of those initial reproductive cells right up to the complex unfolding of entire organ systems.
Okay, let's unpack this then. Before conception can even happen, we need the specialized cells involved. They need to be formed first. This crucial process is called gametogenesis.
Right, and this really highlights a fundamental difference biologically between males and females. How are these unique reproductive cells, the gametes, actually produced?
We'll look at oogenesis for females and spermatogenesis for males, and the distinctions here are, well, they're incredibly important for understanding reproductive health.
Okay.
So for females, oogenesis, that's the production of an ovum or egg. It actually begins incredibly early. We're talking during fetal life itself.
Wow, really, before birth?
Yes, specifically up to about 30 weeks of gestation. And it's a tightly controlled process, orchestrated by hormones like GnRH, gonadotropin-releasing hormone, FSH, follicle-stimulating hormone, LH, luteinizing hormone, and estrogen. This whole intricate dance results in the production of just one mature ovum per reproductive cycle, which averages, about 28 days.
Just one, typically. And it involves meiosis, that specialized cell division, where primary oocytes, starting with 46 chromosomes, undergo meiosis the first. That it forms a secondary oocyte and these tiny polar bodies. Then meiosis the second only completes if that ovum is actually fertilized, and that ultimately yields a mature ovum with just 23 chromosomes.
So it's definitely not a continuous factory line for females then. Starts super early, very regulated output. That seems like a huge difference compared to males.
Exactly. A profound difference. And that distinction carries really significant implications for fertility, reproductive aging, all sorts of things. Now contrast that with males, where spermatogenesis, the continuous production of sperm, kicks off at puberty and just keeps going throughout life.
Continuously.
It's also hormonally controlled, involving GnRH, FSH, LH, testosterone, and even growth hormone. But here's where the difference in scale really stands out. This process is incredibly prolific. We're talking anywhere from 35 to 200 million sperm per ejaculation.
Million compared to one ovum. That's staggering.
It is. And each primary spermatocyte ultimately yields 4 mature sperm. And like oogenesis, it also involves meiosis, reducing that chromosome number from 46 down to 23.
So whether it's that one precious ovum or millions of sperm, what does this reduction division mean for the gametes themselves? What are they actually carrying?
Both sperm and ovum are what we call haploid. Think of it like this. They each contain exactly half the genetic blueprint needed for a new human. Specifically, 23 chromosomes each, 22 autosomes carrying most of our general genetic traits, and 1 sex chromosome.
Ah, the X or Y.
Precisely. That single sex chromosome determines whether the new individual will be genetically male, XY, or female, XX. And structurally, they're different too. The ovum is relatively large, it's non-motile, packed with nutrients to support those very first stages of development. The sperm, totally different, tiny, highly motile with that characteristic tail, and its primary job is just to deliver its genetic package efficiently.
Okay, here's where it gets really interesting. The actual moment of conception, fertilization. This is where those two unique cells actually meet up.
It is. And fertilization isn't just a simple meeting. It's a carefully orchestrated sequence, a kind of microscopic ballet, you could say. It typically happens in a specific part of the fallopian tube, the ampulla. So first, you need ovulation, the release of the ovum from the ovary. That usually happens about 14 days before the next menstrual period is expected.
Okay.
Once released, that ovum is incredibly time-sensitive. It's only viable for about 24 to 48 hours after release, and it's quickly swept into the fallopian tube, basically waiting.
And what about the sperm? We heard there are millions of them, but clearly not all of them can make it, let alone fertilize that single egg.
Indeed. Billions are ejaculated, but only a fraction a really tiny fraction, actually navigate the incredible journey through the female reproductive tract to reach the ovum. Sperm can remain viable in the female tract for maybe up to 24 hours, sometimes a bit longer, sort of waiting in the wings. But there's a crucial step for them before they can even attempt fertilization. It's called capacitation.
Capacitate.
Yeah, think of this as a final power-up or activation step that has to happen within the female tract. It changes the sperm's outer membrane, making it capable of penetrating the ovum's layers. Without capacitation, fertilization just won't happen.
Okay, so you've got a viable egg waiting and capacitated sperm arriving. How does that one lucky sperm actually get in? And how does the ovum stop, a crowd?
Right, good question. The fertilization process itself is quite remarkable. Once a sperm reaches the ovum, it first has to penetrate the outer layer, the corona radiata, and then the inner, tougher protective layer, the zona pellucida. This penetration is helped by something called the acrosome reaction. Enzymes are released from the sperm head, essentially dissolving a pathway through.
Like tiny chemical drills?
Kind of, yeah. Once one sperm successfully fuses with the ovum's membrane, it triggers an immediate and vital protective mechanism. It's called the zona reaction. It's like a chemical lockdown of that zona pellucida. It instantly becomes impenetrable to any other sperm.
Yeah, a very effective bouncer system.
Absolutely crucial. It ensures that the resulting embryo gets the right amount of genetic material, half from one ovum, half from one sperm. Finally, the genetic material, those 23 chromosomes from the sperm and the 23 from the ovum, combine. They form a single diploid cell, the zygote, now with a full set of 46 chromosomes. And this fusion also instantly determines the genetic sex of the new individual.
Okay, so fertilization complete, we have the zygote. But that single cell doesn't stay single for long, right? What happens in these first couple of weeks as it starts its journey?
Not long at all. As soon as fertilization occurs, this pre-embryonic period kicks off. That's the first 2 weeks. It's a time of intense, rapid cell division and organization, all leading up to implantation in the uterus. The zygote immediately begins a series of mitotic divisions, just dividing and dividing. We call this process cleavage. And this is happening while it's traveling down the fallopian tube towards the uterus.
Cleavage. So just multiplying cells. How fast does this happen? What does this tiny cluster become?
It's pretty quick. By about day 4, this rapidly multiplying ball forms a solid mass of, say, 16 cells. We call that a morula. It literally looks like a tiny mulberry under the microscope.
Amarilla, okay.
Then, fluid starts to enter this morula, creating a cavity and transforming it into a hollow sphere. This is known as a blastocyst. And this blastocyst has 2 really distinct and critically important parts. You've got an inner cell mass, sometimes called the embryoblast.
Embryoblast, that sounds important.
It is. That's the part that will eventually develop into the embryo itself, all the body parts. And then there's an outer cell mass, the trophoblast. This layer will contribute to forming the placenta, the vital life support system.
Okay, inner mass becomes baby, outer mass becomes support structure.
Essentially, yes. And the grand finale of this pre-embryonic period is implantation. This is where the blastocyst firmly embeds itself into the wall of the uterus, which is now specially prepared and called the decidua. This usually happens by about 6 to 10 days after conception. The decidua itself, that uterine lining, literally transforms. It develops specialized layers, the basalis capsularis vero, to welcome and nourish this new life, providing crucial support and a rich environment.
So as that tiny, rapidly dividing blastocyst settles into its new home in the decidua, a truly astonishing transformation begins. We're now moving into the embryonic period, right? Weeks 3 through 8, this is where the blueprint gets laid down.
Absolutely. This period is defined by intense cell differentiation and organ formation. The proper term is organogenesis. It's when the basic body plan gets established. And importantly for anyone in healthcare, it's also a period of immense vulnerability for the developing embryo to potential disruptions.
Right, because so much is happening so quickly.
Exactly. That inner cell mass we mentioned. It differentiates into 3 primary germ layers, and each layer is destined to form specific tissues and organs. Understanding these layers is really key to appreciating how every single part of the body is built.
Okay, lay it on us. What comes from which layer?
Okay, so first the ectoderm. Think outer layer.
Yeah.
This forms most external structures and the nervous system. So the entire nervous system, brain, spinal cord, but also the epidermis of the skin, hair, nails, tooth enamel, and even the mammary glands.
Wow, quite a mix. What about the middle layer?
That's the mesoderm. Meso means middle. This develops into crucial supportive and circulatory systems. Think connective tissue like cartilage and bone, all types of muscle, the entire circulatory system, heart, blood vessels, blood cells, the lymphatic system, the kidneys, and the gonads, so ovaries or testes.
Okay, structure, movement, circulation, filtering.
Exactly. And finally, the endoderm. Endo means inner. This innermost layer gives rise to the lining of the digestive tract and the respiratory system, plus important glands like the liver, the pancreas, the thyroid, and the pituitary.
Got it. Ecto outside nerves, meso middle structure circulation, endo inside linings glands.
That's a good way to remember it. And during this embryonic period, weeks 3 to 8, we see incredible milestones happening week by week. It's just breathtakingly fast. For example, by week 3, the neural tube, which is the precursor to the brain and spinal cord, starts to close. And the heart literally begins to partition and starts beating.
Week 3, already a heartbeat. Amazing.
It is. By week 4, the heart develops distinct chambers, and you can see little limb buds, the very beginnings of arms and legs starting to appear. Jump to week 6, external ear development begins. The upper limbs have flattened out, looking more like arms, while the lower limbs are still sort of paddle-shaped. And by the end of week 8, fingers and toes are distinct. That little embryonic tail, which is a normal part of early development, disappears. By this point, the end of the embryonic period, the embryo truly has a distinctly human appearance. All the main organ systems are laid down in rudimentary form.
Incredible. So after week 8, we shift gears again. From week 9 until birth, it's all about growth and refinement. This is the fetal period.
That's right. The fetal period is indeed characterized by rapid growth, the continued maturation of all those organ systems that were established earlier, and increasing viability. Viability meaning the potential to survive outside the womb, though obviously still needing significant support if born very early. It's when the body is really fine-tuning itself, preparing the fetus for life outside. Many systems continue to develop and become fully functional.
So what are some key highlights here?
Okay, well, between weeks 9 and 12, the external genitalia become more distinguishable, so you might be able to tell male from female. The eyelids actually fuse shut for protection. And the heart's activity can usually be readily detected by a Doppler ultrasound device by this point. We also see the beginnings of a sucking reflex.
Okay, getting ready for feeding.
By week 16, the face looks much more distinctly human, and the fetus starts to make more rapid, coordinated movements, though the mother might not feel them quite yet.
But soon. That's often when the mother starts feeling those first flutters, right? That amazing sensation known as quickening.
Precisely. By week 20, fetal movements, or quickening, are typically felt by the mother. This is also when lanugo appears.
Lanugo.
That's a fine, downy hair that covers the body. And also vernix caseosa, which is a protective, sort of cheesy or creamy substance on the skin. Both help protect the delicate fetal skin from the amniotic fluid.
Makes sense.
Now, critically important, especially for neonatal care, is what happens between weeks 24 and 28. Surfactant production begins in the lungs.
Surfactant. I know that's crucial for breathing after birth.
Absolutely essential. It's a substance that reduces surface tension in the alveoli, the tiny air sacs in the lungs, preventing them from collapsing when the baby exhales. Its presence dramatically improves viability if a baby happens to be born prematurely. The skin also appears quite wrinkled and red during this time. And for male fetuses, the testes typically begin their descent from the abdomen towards the scrotum.
Okay, so viability really starts to increase around that 24, 28-week mark because of the lungs.
That's a major factor, yes. Moving on, by weeks 32 to 36, the lanugo usually starts to disappear, the skin smooths out as fat accumulates underneath, and head hair develops more noticeably. The tests are also typically fully descended into the scrotum by the time of birth, and finally by weeks 38 to 40, we're at full-term development. We expect mature lungs, a robust sucking reflex, significant fat accumulation for insulation and energy reserves. Basically, ready for birth and the transition to life outside the womb.
It's just an incredible process, but the developing baby isn't alone in there. It's intricately supported by some truly incredible temporary structures, these auxiliary structures.
Yes, and this really prompts us to consider, how does this amazing developing human get everything it needs, oxygen, nutrients, and how does it efficiently get rid of waste products? The placenta and these other structures are absolutely vital for this continuous exchange and also for protection. First, there's the amnion. This membrane forms the amniotic sac, often called the bag of waters. This sac is filled with amniotic fluid. This fluid does several amazing things. It acts as a fantastic cushion protecting the fetus from external bumps or pressure. It maintains a constant, stable temperature, preventing heat loss. And crucially, it allows for fetal movement. That movement is essential for proper musculoskeletal development. It also allows the fetus to swallow fluid and practice breathing movements, which aids lung development.
So much more than just cushioning. And then there's that physical connection, the umbilical cord. What exactly is it carrying back and forth?
That's the umbilical cord, and it's truly the lifeline. Typically, it contains two umbilical arteries. Remember, arteries carry blood away from the heart, so these carry deoxygenated blood and waste products from the fetus to the placenta for disposal. And vitally, it contains one umbilical vein. Veins carry blood toward the heart. This carries oxygenated and nutrient-rich blood from the placenta to the fetus.
2 arteries away, 1 vein towards. That's easy to remember.
Exactly. And all three of these crucial vessels are embedded in Wharton's jelly. This is a robust, gelatinous connective tissue that provides protection. It prevents the cord from being easily compressed or kinked, which would cut off that vital supply line.
Smart design. And that brings us to the placenta itself. This temporary organ sounds like the command center.
You could definitely call it that. The placenta is a remarkable temporary organ. It's nothing short of a biological marvel with multiple critical functions.
Okay. What exactly does the placenta do? It sounds like it's doing a lot of heavy lifting.
It absolutely is. It's highly dynamic. First, it has metabolic functions. It can actually synthesize substances like glycogen for energy storage, cholesterol, fatty acids. It acts almost like a temporary liver for the fetus in some ways. But its primary and most vital role is transfer. It facilitates the efficient exchange of gases. Oxygen comes across from mother to fetus. Carbon dioxide goes back. It transfers crucial nutrients like glucose, amino acids, minerals, vitamins, and that allows waste products like urea and uric acid to pass from the fetus back to the mother's circulation for her kidneys to eliminate.
So it's the lungs, kidneys, and gut all rolled into one interface.
In terms of function, yes, that's a good analogy. And this exchange isn't just passive diffusion for everything. It happens through various sophisticated mechanisms: simple diffusion, facilitated diffusion, active transport requiring energy, even phagocytosis, which is like cell drinking. That's how, for example, maternal antibodies, like IgG, are actively transported across, providing crucial passive immunity to the fetus before its own immune system fully matures.
Giving the baby a head start on fighting infections.
Exactly. And finally, the placenta has crucial endocrine functions. It's a hormone factory. It produces vital hormones like human chorionic gonadotropin, HCG, that's the hormone detected by pregnancy tests, initially maintaining the corpus luteum. It also makes human placental lactogen, HPL, estrogen, and progesterone. All of these are absolutely essential for maintaining the pregnancy itself, promoting uterine growth, preventing contractions too early, and supporting fetal development.
Wow. Metabolic, transfer, endocrine.
Oh.
It's truly multifunctional organ. Now, something else that's fascinating is how the baby's circulatory system works inside the womb. It's completely different from ours, right? Because the lungs aren't doing the oxygen exchange yet.
That's a critical point, and understanding it is vital for anyone assessing a newborn. The fetal circulatory system is uniquely adapted to bypass those non-functional fetal lungs. The placenta is doing the gas exchange. So the fetal circulation involves specific shunts or shortcuts that redirect blood flow away from the lungs and also partially the liver.
Shunts. Okay, like bypass routes.
Exactly. There are 3 main ones to know. First, there's the ductus venosus. This shunts a significant portion of the highly oxygenated blood coming from the umbilical vein directly into the inferior vena cava, largely bypassing the fetal liver.
So nutrient-rich, oxygen-rich blood goes straight towards the heart, skipping a full pass through the liver.
Much of it does, yes. It allows that highly oxygenated blood to reach the heart and brain faster. Next, inside the heart itself, there's the foramen oval. This is a special opening, a flap, located directly between the right atrium and the left atrium.
An opening between the 2 top chambers of the heart.
Correct. It allows much of that oxygenated blood returning to the right atrium to flow directly across into the left atrium. From there, it goes to the left ventricle and gets pumped out to the systemic circulation, especially prioritizing the brain and upper body. It effectively bypasses the pulmonary circulation, the trip to the lungs.
Okay, so skip the lungs via the foramen oval. What's the third shunt?
The third one is the ductus arteriosus. This is a blood vessel that connects the pulmonary artery directly to the aorta. Any blood that does get pumped from the right ventricle into the pulmonary artery mostly gets shunted through the ductus arteriosus into the aorta, again bypassing the lungs and joining the systemic circulation. This whole system cleverly ensures that the most oxygenated blood is delivered to the developing brain and the heart muscle itself first.
It's incredibly efficient for the intrauterine environment. And then when the baby is born and takes that first breath, this whole setup changes instantly, like flipping switches.
Yes, it's a dramatic and incredibly swift transition, a testament to the body's adaptive capacity. With the baby's first breaths, the lungs inflate, and pulmonary vascular resistance drastically decreases. Air replaces fluid. Simultaneously, when the umbilical cord is clamped and cut, systemic vascular resistance increases because that low-resistance placental circuit is removed. This rapid change in pressures causes these fetal shunts to functionally close very quickly. The increased pressure in the left atrium pushes the flap of the foramen oval shut. The ductus arteriosus constricts in response to higher oxygen levels and changes in prostaglandins. The ductus venosus also constricts. This closure redirects blood flow through the now functional lungs for oxygenation and through the liver for processing, establishing the mature, adult pattern circulation we rely on outside the womb.
An amazing physiological shift. Finally, let's just briefly touch on what happens when there's more than one baby developing a multifetal pregnancy.
Right. And if we connect this to the bigger picture for healthcare, multifetal pregnancies involve distinct developmental pathways depending on how many zygotes were involved. This has significant implications for monitoring the pregnancy and potential complications. The two main types are monozygotic and dizygotic twins. Monozygotic, or identical twins, result from a single fertilized ovum that, at some point very early in development, splits into two separate embryos.
One egg, one sperm, then it splits.
Correct. Because they originate from the exact same zygote, they share the exact same genetic material and are always the same sex. Now, depending on when that split occurs, they might share certain structures. They could share a placenta or the chorion or even the amnion. These variations impact potential risks during the pregnancy, like twin-to-twin transfusion syndrome.
Okay, so identical twins come from one zygote splitting, and dizygotic twins must be different then, not genetically identical.
Yes, completely different origin. Dizygotic or fraternal twins result from the fertilization of two separate ovas by two separate sperm during the same cycle.
Two eggs, two sperm.
Exactly. They are genetically distinct, just like any other siblings born at different times. They just happen to share the same womb. They may be different sexes or the same sex. And critically, because they come from two separate fertilization events, they always have separate placentas, separate chorions, and separate amnions, although the placentas might fuse if they implant close together. We also sometimes see high-order gestations, triplets, quadruplets, and so on. These have become somewhat more common, largely due to the increasing use of assisted reproductive technologies, fertility treatments, and naturally, these pose unique challenges and increased risks, requiring specialized care for both the pregnant parent and the babies.
Fascinating distinctions. Well, we've just navigated the truly incredible journey of human development all the way from conception to birth, highlighting that intricate dance of cells, hormones, timing, and those specialized structures. It's really quite astounding what happens in those 40 or so winds.
It really is. This deep dive just scratches the surface, but it reveals the immense complexity and the incredible precision required for normal development. It's honestly a powerful reminder of how much has to go right, moment by moment, for life to begin and flourish. And for you, as future healthcare professionals, understanding these fundamental biological processes isn't just fascinating background info, it's truly the bedrock. It's the foundation upon which all your future knowledge of patient care, assessment, recognizing abnormalities, and planning interventions will be built. It equips you to understand the normal so you can quickly identify, understand, and respond appropriately when things deviate.
Absolutely fundamental. So thinking about everything we've covered, from gametes to germ layers to fetal shunts, what stands out to you from this deep dive? What new questions does this incredible understanding spark for your own learning journey as you prepare to care for the next generation right from its very beginning?